We have the next talk about somatic and germline mutational signatures that differ in the human mitochondrial genome. And unfortunately, the assigned speaker, Kristina Ushakova, cannot make it, so we have a substitute, which is Dmitry Iliushchenko. Okay, no, here. Okay, go ahead.
Hello, dear colleagues. My name is Dmitry Iliushchenko, and I substitute my colleague, Kristina Ushakova, who couldn't attend to this conference. And I will talk about mutational spectrum of the human mitochondrial genome. Somatic and germline signature differ. We will talk about mutational spectrum. Let's start off with it. Why do we use it, and what is it? Closer? Okay. Evolution consists of two complements, mutation and selection. Mutation that appear in a genome could be selected or not selected, could be purified or supported by selection. And every mutation is characterized by probabilities with which nucleotide transfer into each other. That probabilities combined generate specific unique signatures. That signatures, it's normalized matrix, what we call mutational spectrum. For different purposes, we could use different mutational spectrum. This is two-component mutational spectrum with only transition and transversions. If we take every letter and every letter is valued again to a larger, we will get a 12-component mutational spectrum. And if we will add neighbors to these letters, we will get 192 mitochondrial component spectrum. And our goal, our main goal, is compared germline and somatic line mutational spectrum. And we use two data sets. One of them is from cancers, from the article, from the paper from Natural Genetics. And from the germline, we reconstructed phylogeny. This is our hard pipeline. I will not talk about it. This is really hard. It's huge. But I want to say that special thanks for Chernych Igor. And we got almost 1 million mutations. Then we just took only 4-fold degenerative mutations. And let's talk about our results. Let's first look at the 12-component mutational spectrum. In the bottom, you can see germline, on the top, somatic line, and transitions are reproduced by faint and bright colors, and transversions are gray. And what we can see here, first, the most common substitutions, as we know, is C to T and A to G. And we can go deeply and add some neighbors for this substitution, and we will get 16 bars for each substitution. And we can see here that's equivalent C to T on a heavy chain, G to A on a light chain. It's not really much up. And we think this is some asymmetry between two equivalent substitutions. What we can see here, we have a ratio for C to T and G to A for soma and germline, and we see here that somatic line has more level of asymmetry compared to the germline. What about A to G to C to T? T to C. Somatic line also has more level of symmetry. What we can say about that, that germline mutational spectrum is less asymmetric, less chemically damaged. That means more protected. And let's take a look at 196 component mutational spectrum. Let's remove all transversions and compare colors, faint and bright, and we'll get this great graph. And what you can see here, the first letter, it's always different. Second, it's our substitution. And the most common third letter in somatic line is G. And on a germline, it will be C, how we can see here, on a C to T and A to G both. And why more C to T in soma, CPG in somatic line? We are not the first who used cancer data to make mutational spectrum, but there is only 96 mutational spectrum, while we use 196 mutational spectrum. And here we can see that our data is in line with this data, but authors of these articles don't really go deep into details and don't talk about why is it, what is the signature of these substitutions. And we look at this cosmic data, SBS1, single base substitution, and how we can see our results really compare to these results, and the most common substitution C to T, and the most third letter, it's G, like CPG in somatic line. And they correlate this substitution with aging. And higher C to T in context of CPG profile in somatic line, it's a signature of age. Let's talk about CPC in a germline. And we wrote a lot of paper about this. Why is it so? And this is a really great paper of Dmitry and Paul Deutsch, and this is about redox stress in yeast in a single strain DNA, like in a heavy chain of mitochondria. And how we can see here, the most common substituted C to T with context of CPC. And they correlated with redox stress. Also, they tried to compare mitochondrial DNA with context, but they didn't get normal results because they used somatic lines, not a germline. And the last conclusion, higher C to T in context of CPC in germline, it's a marker of redox stress. And this is our take-home message. I want to say thank you to Christina, who prepared this great project, and I want to say thank you, my lab, Mitochondrial Functional Genomics, and especially our supervisors, Konstantin Gunbin and Konstantin Papadin. Thank you for your attention, and I'm ready for your questions.
Thank you. It's perfect timing, and we have time for questions, really.
Just a quick clarification. So there is very strong strand dependency in mitochondrion, and there is very strong strand dependency in mitochondrion with respect to replication direction. So I don't know if you took this into account in any way in the analysis.
Sorry, could you?
There is very strong dependency in mitochondrion in mutation characteristics on the strand, on DNA strand. Oriented with respect to replication direction. So I don't know if you took this into the analysis or you combined directions, you combined mutations on both strands.
We just use, thank you for your question, we use a high straight of mitochondria. This is like a single strand on the east, like I told. Thank you.
Okay. Igor.
So you interpret your difference between the germline and the soma as the difference in the mutation rate, but can it be due to selection being stronger on the germline?
This is really great question, and what I would say about somatic line, we just, if we see here this mutation in spectrum, they are not selected, and this is really great. What about some germline? We use just 4-fold synonymous for degenerative sites, and we expect that there is no selection against all these substitutions.
Are there any signs of uneven codon usage in mitochondria?
Yes.
No, that's a question.
This question?
Yeah. Well,
yes, we used a special.
so there is
mitochondria.
some selection on synonymous sites as
Yes.
well.
Yeah, we use some difference, and this fourfold, especially for mitochondria, of course.
Okay, well, then that's it. Thank you.